Does everyone see the slideshow? Okay. All right, just checking. Always have a little bit of housekeeping, even though it's been working for us. Okay, so we left off yesterday talking about how parietal cells secrete hydrochloric acid and the exact mechanism, exactly what's going on. And I went into great detail a couple times about this. So just as a reminder, this particular slide is extremely important. I would memorize all the different details I went through yesterday on this, okay? Particularly how do we get from the bloodstream, how we get starting reagents such as carbon dioxide and water, and how do we end up with the carbonate ion, as well as um, protons, which will then bind to chlorine to form hydrochloric acid. Okay, so that whole process you need to know. Okay, all right. So let's talk about how pepsin actually digests protein. Like what's going on. Okay, and I kind of already mentioned this yesterday. All right. So we all know that pepsin is the active form of pepsinogen. Okay. Pepsinogen is the inactive form of pepsin that's secreted by our chief cells. Okay. Inside our the gastric pit. Okay. Now it gets converted into pepsin with when hydrochloric acid is making the environment more acidic. So remember, hydrochloric acid is going to reduce the pH. Okay, reduce the pH, and this is going to cause pepsinogen to be converted into the active form of itself, pepsin. Okay, and usually you can see here that the reason why it's because of this section over here. This section over here is going to get cleaved off. Okay, so this peptide here gets cleaved off with the help of the acidic environment caused by hydrochloric acid, and once this peptide gets cleaved off, okay, it's going to allow pepsin to be activated. So this means that this piece of peptide here, okay, this orange peptide over here, has an inhibitory function on pepsin, okay? It what it does it's going it likely it has to do with steric hindrance. So that means that the way that pepsin works to break down its target proteins, it binds to it. So there's certain binding sites on the protein. So for example, over here, we have pepsin, okay? So pepsin is the activated version of this enzyme, okay? It's going to target a specific binding site on the dietary proteins, okay? And it's going to allow it to be digested, okay? Now, this little peptide here, this orange peptide, what it does, it interferes sterically with the function of pepsin, okay? That means that let's say pepsinogen is lurking around, right? It's going to try and attack its target proteins to break it down. It's not going to be able to actually bind to it and actually do its effect or function on it. We're chopped up into smaller pieces, okay? Because this piece here is blocking the full function. There's steric hindrance, meaning spatial interference caused by this little peptide, okay? And this whole entire this whole entire process functions in a positive feedback loop, like I mentioned yesterday. Okay. Once you activate some pepsin, the pepsin itself goes back to activate more pepsinogen. Okay. So the pepsin itself, okay, just erase this, too clustered. Okay. The pepsin itself, okay, it's going to go back over here and activate more pepsinogen into the pepsin form. Okay. So working both ways. You're going to have the hydrochloric acid converting pepsinogen to pepsin, and then pepsin itself will also convert pepsinogen into more pepsin. All right. And of course, it's going to break it down to smaller pieces of protein. Okay. Is it so far? Now, this just came. I want you guys to know this. I keep bringing it up every now and then, and I think it's a good thing to understand about form fits function. When you talk about the anatomy and physiology of a human body, okay, these parietal cell and chief cells are they're kind of close to one another. This is really important. The proximity between the two of them are pretty important. Okay, because you want to be able to have this you want this whole process to kind of happen fluidly. Not you don't want to have it take too much time. Okay, our body works pretty efficiently in that manner. Okay, just a little uh, side note, right? Okay, so let's see what happens in okay absorption of nutrients. Now, I one of the hormones I mentioned already, or two we already know. Okay, gastrin and secretin. Okay, now I mentioned to you what secretin does. It's going to help to prevent reduction of the pH. Okay, and we're going to talk about what it exactly does. A little bit more detail, just a tiny bit more detail. Okay, and I promise, and I told you yesterday that we will see it again just briefly. Okay, now of course we have gastrin. Okay, and we know that, okay, so it's going to be secreted by the stomach and duodenum of the small intestine, okay? And it's going to be stimulating pancreatic juice secretion, okay? Then we have cholecystokinin, okay? So, or CCK, okay? And the exam, guys, if I ever ask you something about this, write out the entire name, okay? Don't put CCK. I mean, I'll know what it is, but just for out of habit, you should write the whole entire name out, okay? Um, particular hormone is going to be secreted by duodenum in response to fats and acids arriving from the stomach. Okay, remember your duodenum is the very first section of your small intestine, right? Right as your all the chyme exits your stomach, it's going to enter that area. Okay, and what this CCK does, it's going to stimulate bile and pancreatic juice into the duodenum. Okay, right. And last but not least, secretin. And we already spoke about secretin, okay? It's secreted by the small intestine, specifically your duodenum of the small intestine, okay? And it's going to be a response to the acidity caused by the chyme. Remember, chyme is pretty acidic, right? It's the acidic soup that I keep bringing up. In that analogy, the acid soup, that's what chyme is, okay? Because it's mixed in with a lot of hydrochloric acid, water, some bits of proteins, different foods that you partially broke down, okay? And that acidity is going to activate uh, to secrete secretin, okay? And of course, what secretin does, like I told you guys yesterday, is going to prevent further reduction of the pH, okay? And the way it does this is that it stimulates secretion of bicarbonate, okay? Now remember, bicarbonate is part of the buffering system, okay? So that means that when things get too acidic, it's going to try and bump up the pH back to normal, okay? So therefore, pH of the intestine, okay? Because if it maintains the acidity as it's exiting the stomach throughout our entire gastrointestinal tract, okay, it's gonna burn. It's gonna harm us. All right, make sense, guys? I want you to understand. Oh, because I can ask a not super critical thinking question, just kind of putting one and two together. Okay, if I tell you, like for example, patient A, right, is suffering from some minor ulcers in the upper small intestine, okay? Or just not even the upper small intestine, just throughout let's say throughout the intestine, okay? Specifically the small intestine, okay? What's a possible reason why this person's getting some ulcers? Knowing what right now, okay, from what we just went over. And that's a bacterial infection. Yeah. Yeah, Caroline. So this person may have an issue um, producing secretin. Yes. Of course, the alternative will be bacterial infection. But I told you guys, we know that the person doesn't have a bacterial infection. Now, if I told you guys that there's a suspicion there might be a bacterial infection, then you would say like maybe Campylobacter jejuni, some kind of gastrointestinal bacteria. All right, maybe um, O157H7 potentially can do this, but usually like Campylobacter are known for this. Okay, to cause intestinal ulcers. That's what I want to mention on this. Oh yes, the bullet point on the top. Okay. Now the gastric and intestinal hormones, all these collectively, I mean, this is gonna be reiterating everything I just said. Okay, it's gonna be stimulating bile and pancreatic juice secretions into the small intestine. Okay, so that's one of the functions of all these hormones. All right. And we can specifically CCK does this, okay, and gastrin. Uh, so just remember what all these three do, okay? It's pretty straightforward. Of course, these are still going to be regulated by our vagus nerve, okay? They're all regulated by the same cranial nerve, okay? All these different hormones, all right? Okay. Everyone clear on that? I'm concise to the point as much as, I'm rambling quite a bit still. All right, let's talk about our test and secretion motility. So this is going to be, kind of a little bit more dry. There's no pictures on this particular slide. So, what do you want to know from here? Okay. Well, you want to know what exactly how much absorption and secretion occurs in the small intestine. Okay, and what exactly is going to happen in the small intestine. And there should be a picture after the slide. And this specific slide won't have it. Now, secretion. Okay. The amount, and we're going to be doing about 1 to 2 liters of intestinal juice per day. Okay, and most of it is going to be water, some mucus, and some bicarbonate. Okay, as well as we're going to be secreting in response to intestinal distension, hypertonic, and acidic content of tryma arriving from the stomach. Okay, all intestinal juice is going to be secreted due to our the acidic environment and the hypertonic environment caused by the chyme in the stomach. Okay. Let's talk about intestinal motility. Okay, so we know there's a few things happening. Okay, primarily we have smooth muscle, right? The smooth muscle will be very similar to what we've been following from as soon as we entered, as soon as the food enters our mouth and you swallow it. We have the peristaltic movement. The longitudinal muscles, longitudinal smooth muscles that wrap around these different organs. All right. So this whole entire thing continues, and we mentioned this when we first started the chapter that this peristaltic movement, this contraction of the smooth muscles, occurs from the top to bottom. Okay, so in the entire system, we're gonna have this peristaltic movement. Okay, and to help it move time towards the large intestine. Now, I'm going to tell you right now, the way that the chyme is going to be moving in the small intestine, it doesn't just simply move straight through it, okay? It actually moves kind of back and forth, okay? It moves kind of back and forth in your small intestine, but there's a net movement towards the large intestine, okay? Now, the reason why you're going to have this kind of back and forth movement of chyme is to further mix up and break down the chyme into smaller components for our small intestines to reabsorb all the nutrients and water from that point. Okay, because remember, a major reason function, I should say, function for our small intestines are to reabsorb all the good nutrients. Okay, as well as water. All right, and that's where all of that, all the remaining matter becomes fecal matter eventually, right? When we remove all the nutrients from chyme, okay, it becomes fecal matter. All right. Now, there's going to be peristaltic movements, okay, as well as segmentation, okay. Now, the segmentation is going to be the way, the shape that our small intestines look like with the way the muscles are, okay? And this is going to cause this back and forth movement of whenever the muscles contract on our small intestines, the segmentation nature of the small intestine is going to allow for back and forth movement, which is going to increase churning, okay? Now we'll see exactly what I mean right now. Okay, so here we go. All right. So segmentation. Okay, and this is only occurring in the small intestine. All right. Now we have this. These are caused by little circular muscle contractions, and these little circular muscle contractions, and I'll just circle them right now. Actually, so is over here like this. Okay. Now what happened is between over here, over here, over here. You're going to have contractions of each segment, okay? Each segment is going to have a mini contraction, and this mini contraction is going to allow the chyme to further break down and mix, okay? And it's going to be moving the chyme back and forth, as you can see here, right? It's going to be moving this way, then this way, as you continuously contract. But the net movement of a chyme is this direction, Okay, we're gonna go towards the large intestine, right? And first, now remember, okay, just to be clear, guys, okay, segmentation is only in the small intestine. Peristalsis is everywhere. It's both the small intestine as well as the large intestine. Okay, so your colon and your small intestines will have peristalsis contraction. There's still smooth muscle there. Okay, remember the fecal matter eventually, once it enters your large intestine, has to reach the rectum. Okay, the rectum is the only muscle and your GI tract that you're able to actually contract voluntarily. Okay, uh, just be clear on that. All right, makes sense. So the patient contractions, remember, are mini contractions, circular contractions that happens towards the middle of each segment. Okay, and each segment it's separated by this little kind of like pinch. All right, guys, but it does move back and forth. Okay, I don't actually state it out outright over here, but there is a back and forth movement because of the churning that happens. Okay, so if I ask you on the exam, you know, true and false, the movement of chyme through the small intestines happens in one direction only. And you say that's false because it happens in back and forth. However, the net movement goes towards the large intestine. Okay, so be clear on that. All right. So accessory glands secrete fluid and enzymes into GI tract. Okay, so that's just the title of this section. All right. So let's talk about the liver and gallbladder and what exactly is going to be happening. Okay. All right. So now, the one thing I want you guys to understand about the liver, okay? The cells in the liver are called hepatocytes. And I actually do have it down here, okay? Hepatocytes are the type of specialized cells in the liver, okay? And these are regenerative. Remember, the liver is actually pretty regenerative, luckily for us, okay? Now, one of the and this is particularly useful because we only have one liver, right? It's not like our kidneys where we have two of them. We could, you know, God forbid, lose one, right? Or you may have to donate a liver to a kidney to somebody, or but anyways, what okay. The liver manufactures bile, okay? And let's talk about it a little bit, okay? So the gallbladder itself in the liver is going to store bile to be secreted into the duodenum, okay? So the bile will be secreted into the duodenum. It'll be the first, and then we all know this is the upper portion of the small intestine, right? And then we have this bile itself is a yellowish green fluid, and this has minerals, cholesterol, fats, phospholipids, pigments. Okay, specifically, the pigment is bilirubin. Okay, we have bilirubin. Okay, and we have hemoglobin decomposition. Okay, and bile acids as well. Okay, so I want you to know this. All right. Now, the function of bile is going primarily to emulsify fats. Okay, and emulsifying fats is the process by which we're going to break down fats physically. Okay. However, the vast majority of this is going to be reabsorbed. Okay, in the ileum, and only 20% is lost in feces. Okay, so we have a pretty high reabsorbing rate of this of bile. Okay, uh, is about 20% feces. Now, the liver, okay, is going to filter. That's the primary function of the liver. I'm not going to go too much into the liver. There's only a few slides, really. It's the primary function is to filter blood out and detoxify toxins in blood coming from the small intestine. Okay. Now, guys, what's the other organ that detoxifies blood? What's the other organ of this? There's another organ that does this. Yeah, the kidneys. Yeah, you need to know that. Okay. The liver and also the kidneys, they both, one of the functions is to detoxify toxins, okay, coming from the blood. However, the kidneys, we're not looking at the small intestine, the blood coming from the small intestine, okay? All right. Now, like I said before, the hepatocytes are cells of the liver. Okay, it has cytochromes that chemically break down toxins. So the cytochromes themselves are what actually breaks down the toxins. That's the compound that will the molecule that will actually break down toxins chemically. Okay. Now remember, chemical breakdown of toxins means that we're converting the toxins from one form into another. Okay. So for example, uh, let's talk about catalase. Okay, so catalase is an enzyme that converts hydrogen peroxide into water. Okay, now hydrogen peroxide is actually one of the byproducts of cellular respiration. All right, aerobic respiration. Okay, and we need catalase enzymes to push forward the reaction to convert hydrogen peroxide into water because hydrogen peroxide is pretty toxic. Okay, so this chemical conversion from hydrogen peroxide into water is an example of a breakdown of toxin. Okay, you're basically taking a toxic compound and convert it into something that's not harmful, which is like something like water. Usually, water is usually a byproduct of this kind of breakdown. Okay, now this and we don't really look at because I do kind of want to talk about it since this you guys are in nursing, right? Uh, Oh, here, there's a picture over here. Okay. Okay, I should go around over here and then. I do want to talk about it a little bit, just some kind of disease. So, guys, what's a disease, okay, or infection, I should say, that is famous for infecting your liver? And I want you guys to take note of this, okay? Just it's not gonna be too complicated, right? Okay, hepatitis, right? Exactly. So, hepatitis is a very famous viral infection, okay? So remember, hepatitis is a virus, right? Now, how many types are there, if anyone knows? Is that only, yeah, there's three. There's A, B, and C, okay? And the virus that causes each of them are the name. So the hepatitis A virus, hepatitis B virus, and hepatitis C virus, okay? Now, all three of them are pretty nasty. Okay? They're really good to have. However, the one people get by contaminating needles, when they go in the clinic get some dirty needles, that's hepatitis C, okay? And hepatitis C specifically causes inflammation, all right? And it can cause a lot of internal bleeding potentially, okay, at your liver, around your liver, okay? Now let's talk about A. Now, hepatitis A is primarily it's hepatitis A specifically is pretty rare. It's typically only contracted by when you consume some contaminated food, okay? But you get some fever, that kind of thing. All right, you're gonna get abdominal pain with that. Now, hepatitis B, on the other hand, is a little bit different between the other two. B is going to be contracted between, it's going to be transferred between body fluids, such as having sex, like sexual transmission, right? That's going to be a classic method of transfer for hepatitis B. Okay, it's more of a sexual, body fluid oriented version of the infection, but it does afflict abdominal pain. You're gonna have some fever. Okay, although the most serious one out of the three, one of the most serious is hepatitis C because it could cause internal bleeding. Okay, now all of these guys can be prevented with the help of a vaccine. That's why when you get your immunizations growing up today, you get shots for various hepatitis viruses. Okay, is everyone clear? I want you to know that. Okay, just this bit of clinical application, right? Maybe some diagnostic a question I might ask on the exam. Okay, I do want to kind of make a few questions like that this time. I just think it's good for you guys to think about it. It helps you to put that you in that mindset a little bit. Uh, anyway, people when you grow up, you usually do get shots. usually do get shots, vaccines. Yeah, there's if you get vaccinated for hepatitis, you'll be protected. I mean, you should know that. Just for everyday life too, just for a little bit of common sense. You should know that, you know, the more things you get vaccinated for, the better. There's only I do understand that there are only certain things you're required to get vaccination for legally, especially if, for example, I know Caroline works in a clinic, right? You're working, Caroline, you work in a hospital, if I'm not mistaken, correct? So, who work in the clinic, who work in labs, like myself, right? I was working in a lab. People who work in these kind of areas, they are required to take additional vaccines. Okay, and they're pretty serious about that because when you work in the clinic, you want to minimize. I mean, you're already working in a pretty hostile environment towards you, right? Because you're gonna be around people who have a lot of different things. You're gonna be around chemicals for scientists, especially biochemical scientists. Um, but they require you to take a lot more than what you normally would get, outside of that field. Caroline, you were required, right, to get additional vaccines, right? Or at the very least, they were pretty thorough with checking your immunization record, correct? Yep. There you go. Yep. Of course, the flu every year and hepatitis B. Yep. Although you know, don't go by what the never vaxxers say. Those are idiots, right? The people who believe that that vaccines cause um, OCD and ADD and all that kind of jazz. They're just morons that are just looking at bogus research done by bogus people that did bogus science. Okay, vaccines are fine. They're safe to use. The only thing that might happen once in a while, you might get some minor version of the virus, right? A very minor version that'll just pass in a couple of days, maybe a little bit of nausea. Okay. For the most part, they're good. The only thing that might happen in the context of the influenza virus is that the cocktail they give you for the influenza virus might not have the right mixture that's predicted. Because remember, just a little side note: the influenza virus, the way they prepare the vaccines for those, they do a genetic uh, guess on the next batch of how the virus is going to mutate a little bit. Okay, and they make guesses based on the antigens, and that's why usually the good influenza vaccines is a cocktail of three different types typically. Um, so they'll give you a cocktail shot that it should protect you. Like I think it was last year where the that annual batch of vaccine were crap. They weren't good because it didn't protect you against the nasty strain of influenza. Yeah, yeah. 1.5. I'm that is. I here. I just don't know what that number is. Uh, so liver. So again, look at the slide right here now. Okay. Oh, it's Drew. Okay. I don't know. It's being WebEx. I don't know what to say. So liver detoxifies your blood coming from the stomach and small intestine and synthesizes bile to emulsify fats. We know that, right? So break down fats, right? Now, of course, we all know it's located, right, everyone? This is going to be reviewed from anatomy. It's located right at the lower end of your rib cage, right? And right underneath, as when you look at the dorsal side of the liver, okay, the posterior side of it, you have the gallbladder. And you remember from the anatomy lab that it's signified by this green colored ball that's in the bottom end of your liver, right, on the back side of it. So that's where it's at, okay? And where all that magic happens. And of course, it's going to be adjacent to the inferior vena cava, and that's where the blood is coming from. The inferior vena cava is going to be delivering all this blood that has all the toxins, and it's going to be dropped off at the gallbladder. Okay, or the gallbladder, the liver, I should say, collectively speaking. Okay. Guys, when you look at it at the anatomy of it, you see how everything's close to one another. How there's a blood vessel over here that's going to be able to drop off all the goods into the liver. Because it's really important everyone understands the how the anatomy works with the physiology, right? Everything has to kind of fit form, for it to work. You can't have the gallbladder exist near your brain, for example, right? I'm making a stupid example, but you can't have your gallbladder exist at where your cranial area is if it's going to have the same function it has in real life, right? Makes everyone what I'm getting at. I met that kind of theme. Okay. All right, so let's talk about pancreas, and I think we'll stop here. This will be the last thing we'll talk about. Okay. So what happens in the pancreas? Well, pancreas is gonna be secreting a lot of different kinds of hormones. All right, and there's two types of secretions. Okay, exocrine and endocrine. Okay. Now, in the exocrine, we have the acini part. It's gonna be the acini part of the pancreas. I don't. Do I have a picture of the pancreas? I have to check. It's going to be secreting called zymogens, okay, or zymogens, however you want to pronounce it, okay. And one of the enzymes, and one of the proteins that's going to be secreted it's called trypsinogen, okay. Now, trypsinogen sounds a lot like pepsinogen, okay, and there's a some similarity between these. Like you could draw a parallel between it, okay. Both of these are going to be the inactive form of the actual protein. Okay, so trypsinogen is going to be the deactivated form of trypsin. Okay, now trypsin, just like pepsin, is going to be digesting proteins. Okay, they both function to digest proteins. All right, now the big difference between the two, okay, is that trypsinogen is activated by enterokinase. Okay, it's an enzyme. All right, now, and the pepsinogen, on the other hand, is going to be activated by the acidity caused by hydrochloric acid. Okay, here it's being activated by another protein, another enzyme. Okay, it's a kinase enzyme. All right. And let's talk about what happens. And here's some other enzymes. Okay, we have chymotrypsinogen and then procarboxypeptidase. Okay, these are two other enzyme examples that will also be converted into its active form. So you have chymotrypsin, which also digests more proteins, and carboxypeptidase, which also digests proteins. Okay, and these are different kinds of proteins that they're all going to be working together to digest collectively. Okay, another exocrine portion of the exocrine system in the pancreas. Okay, you have pancreatic amylase. Okay, and pancreatic amylase is an enzyme that digests starch. Okay, and then a pancreatic lipid, lipase for lipids. Okay. Now, amylase is referring to the way the polysaccharide formation of starch. That's what amylase is going to be breaking down. Okay. And lipase, of course, is in the name of it. We kind of went over it a few times with the naming. It's gonna be for lipids. Okay. It's gonna break down some lipids. All right. So these are all different kinds of different kinds of proteins and enzymes that are going to be secreted by the exocrine portion, the exocrine portion of the pancreas, but specifically the acini part of the pancreas. Okay. Now, in pancreatic ducts, okay, this is the pancreatic ducts. It's going to be part of the exocrine system portion of the pancreas. Okay. Now, the pancreatic ducts, on the other hand, is going to be secreting sodium bicarbonate. Okay. Now, remember, this is going to be something that happens with the help of secretin. Remember, secretin is going to help to release sodium bicarbonate, right? And sodium bicarbonate is going to be an alkaline solution it's a buffer, right? It's part of the buffering system. It's going to neutralize the acid in the chyme, meaning that it's going to increase the pH back to about 7, right? Around 7-ish. All right. That's the whole purpose of the pancreatic ducts. It's going to allow the pathway to secrete sodium bicarbonate in the presence of secretin. Okay. Last but not least, okay. Now the endocrine is going to be the other portion of the pancreas, the other system, and it's going to be within the islets of Langerhans cells. Okay. Now the islets of Langerhans cells are going to be secreting insulin and glucagon. Okay. We're talking about that next class. All right. So insulin and glucagon is going to be regulating our blood sugar levels. All right, guys. This is a little bit tidbit, a little preview for Friday. All right. So we'll stop here. Okay. I apologize for what happened with the that thing with WebEx.